Hi, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. Today is lecture 4 on ion transport. We will be discussing ion transport mechanisms, both the driving forces and the actual molecular components the transporters that transport different ions and substances across a membrane. In the previous lecture, we talked about water transport, and we talked about the driving forces for water transport, which involved the water potential, or basically the chemical potential. And this will be no different. In this lecture, we will be discussing the chemical potential for these ions and determining whether they're passive or active transport mechanisms. It's important to understand the chemical potential. If you don't understand the chemical potential, then it's important that you go back to my lecture 2, where we talk about the principles of the chemical potential and why it's important for the movement of ions. In the second lecture, I'm dividing this lecture into 2 parts because it's so long. In the second lecture, we will talk about the pathways of transport and how ions are transported across a plant or up through the plant. This is not only important for the plant, but it is important for us humans who eat plants and need those minerals for our basic requirements.
Normally when I teach this class, the next lecture is the mineral nutrition of plants, but I've already made a video on that, and you can find that video under this title here called The Mineral Nutrition of Plants, Part 1, The Basics. So to get a background of mineral nutrition, I recommend you see that video first before watching this video. At any rate, let's refresh our general knowledge about mineral nutrition of plants. First of all, plants principally take up minerals through their roots, not through the leaves, although some nutrients can be absorbed in the leaves. The minerals must be dissolved in the soil solution before they are taken up as ions. So the minerals themselves are not directly taken up, or the crystals are not taken up. They're taken up as dissolved minerals, as ions in solution. The roots effectively mine the soil, so the nutrients don't go so much to the roots as the roots go to the nutrients. And therefore, roots proliferate in zones of high fertility and moisture. So we have a particle here where there's an ion exchange going on with negative charges on the soil from the clay. And these ions, the cations, are bound to the negative charges. The anions, on the other hand, are not. They're freely around in solution, as are the potassium and the calcium and the sodium and other cations. They are in an equilibrium with the solution and the soil.
So in this lecture, we're going to focus on short-term transport, how they happen, why they're selective, and how they differ in different plants.
So the major control point for ion uptake into the plant is at the plasma membrane of root cells. Uptake of ions into a root or into a cell is a function of influx and efflux, the inward movement and the outward movement of ions. So transport goes in both directions, and net uptake is the sum of both the influx and the efflux. We must use the chemical potential to understand ion uptake. Does ion uptake require energy? We will need the Nernst equation to determine active versus passive uptake. As you recall, active uptake is based upon consideration of the free energy of the ion. This is a thermodynamic definition that we went through in previous lectures. So the reaction is passive if the ion moves in a downhill direction to a place where there's less free energy or a more negative free energy. The reaction is active if the ion is transported across the membrane in an uphill direction to a place where it is in a higher state of free energy, to a more positive free energy. In an upper example, we see 2 compartments. A chemical potential is shown in compartment A The chemical potential is shown in compartment B, and both compartments are separated by a semi-permeable membrane. Mu is the chemical potential symbol, and mu A is for compartment A, and mu A sub J, the J is referring to some specific ion or substance in this case. The size of the box represents the size of the chemical potential. So in the upper portion, we see a large chemical potential for J in compartment A, and it moves downhill or in the direction of mu J sub B because the chemical potential is smaller. This is a passive transport. It happens spontaneously by diffusion down its chemical potential gradient. In the middle section, we can see both boxes are of the same size. So this means that both chemical potentials are equal and there is no net transport. As a result, we have no active transport under these circumstances. The arrows are going in both directions in equal ways. And in the bottom section, we see a smaller box in compartment A versus a larger box in compartment B with transport moving in the direction of B from A to B. This is then representing an active transport where transport of molecule J or ion J is being transported against its chemical potential gradient. So what are the chemical potential forces that affect ion transport?
Do you remember the four forces that affect the chemical potential of a substance? What are they? I asked you to memorize them. There are four of them. There's concentration, there's the electric potential, which we'll consider here when we talk about ion transport, but the two others are pressure and gravity. And because of the short distance transport mechanisms that we're talking about, they have no influence on the short distance transport of ions. So we don't have to consider them here.
We know that free energy is measured by measuring the chemical potential mu, and mu is influenced by a concentration gradient based upon diffusion and an electrical gradient based upon an electrical potential. Going back to the electrical gradient for a moment, part of the electrical gradient is set up by the diffusion potential, as shown here. How is the diffusion potential created? What is a diffusion potential? We have two compartments, compartment A and compartment B, with initial conditions of a potassium chloride concentration much larger in compartment A and a much lower potassium chloride concentration in part B, separated by a semi-permeable membrane. Those ions, potassium and chloride, will start to diffuse across that semi-permeable membrane from the high concentration compartment A to the low concentration compartment B. However, potassium moves much more rapidly across the membrane than chloride. This is true in real life situations where potassium has a permeability that is about 10 times higher than that of chloride. And as a result of this, we get a charge separation. As more potassium crosses the membrane, the compartment B becomes more positively charged. As a result, the compartment A becomes more negatively charged since it has more chloride ions in it. Over time, the potassium concentration will reach equilibrium before chloride, but eventually the chloride will reach equilibrium and there'll be no net charge separation between the two compartments, and the concentrations of both ions will be equal on both sides.
So the diffusion potential is the separation of charges across a semi-permeable membrane set up by the differences in the permeabilities of ions across that membrane. In our case, cations are more permeable than anions, creating this separation of charge.
So we refer to the chemical potential for ion uptake as the electric chemical potential to emphasize the influence of both the electric and the chemical concentration forces. So the Nernst equation can be used to determine if transport is active or passive. Determining if transport is active or passive can aid us in defining the mechanism of transport. And this, you'll see, is quite significant in terms of our advancement in plant physiology. So to start with, we need to define what the chemical potential is.
So, as I've said before, equations are very useful in summarizing the basic forces that control a certain situation. So we're going to derive the Nernst equation so that you have a better understanding of the forces involved in transport of ions across a membrane. This equation will become very useful in understanding active and passive transport and being able then to identify the molecular mechanisms by how ion transport is occurring in a particular situation.
So the chemical potential, mu here, is based upon mu star, which stands for the standard chemical potential, plus RT times the natural log of the concentration of the ion, plus ZFE, where Z is the charge of the ion, F is Faraday's constant, and E is the electric potential. R is the gas constant, and T is the absolute temperature in Kelvin. So the Nernst equation is derived by setting up a comparison of two compartments at equilibrium. So on the left-hand side of the equal sign, we have a subsymbol I for inside, and on the right-hand side, we have a subsymbol O for outside. And so since we are at equilibrium, mu star plus RT log of CI plus ZF E sub I is equal to what's on the outside as well. So we can rearrange this equation in a way where the mu stars drop out by moving them onto one side, and we end up with ZFE sub I minus ZFE sub O equals to RT log of CO minus RT log of CI. And we can simplify this by combining like terms so that we have ZF times EI minus EO, which is equal to RT times the log of the concentration outside divided by the concentration inside. This is a logarithmic maneuver, and if you're not familiar with logs, I'm sorry, but this is how you can simplify the equation. We bring the ZF over to the right-hand side of the equation now, and we have EI minus EO. So that's the electric field in the inside minus the electric field on the outside, which is equal to, and again, like combination of like terms, we bring over the ZF. We have RT over CF times the natural log of CO divided by CI. The EI minus EO is the Nernst potential, as we'll talk about in a moment. And RTZF times the natural log can be converted to 59.2 divided by Z, which is the charge of the ion, times the logarithm, not the natural logarithm, of CO divided by CI at 25 degrees C. So we'll always use this as an example where we can just easily use a constant 59.2 to make these calculations, assuming 25 degrees. So how do we measure the electric potential? The electric potential can be measured with a voltmeter. We can take a microelectrode and put it in solution. This is our reference electrode. That will be at 0. So if we were to take the second electrode and put it in the same solution, we would have a voltage difference of 0. We can take that second electrode, though, and it's a microelectrode, and like what we did for measuring pressure potential, but this time it's a voltage microelectrode, we can puncture a cell, cross the membrane, and measure any voltage differences between that cell and the reference solution. Here in the upper right, you can see a closer view of that, where that tip has to cross the plasma membrane. And in fact, there is a voltage difference between those. There's a negative voltage difference, and I'll explain that in a moment. Now that we've define the Nernst equation, let's look at an example of how it can be used. Let's take a cell shown here on the right side. It has a concentration of potassium inside of 100 millimolar potassium and an external concentration of 1 millimolar potassium.
This example represents real-life concentrations of potassium inside the cell and outside the cell.
So if we take those concentrations, we can calculate out what the membrane potential should be at equilibrium. Using these concentrations and the Nernst equation, we can calculate out what the membrane potential would be at these equilibrium concentrations. So to do that, we'll substitute in the concentrations into our Nernst equation, and we get the value 59.2 divided by 1, because potassium has a charge of +1, times log of 1 divided by 100, the outside divided by the inside concentration. And if you make this calculation, it's equal to minus 118.4 millivolts at 25 degrees centigrade. Again, under these conditions, no net transport of potassium would occur because the chemical potential difference is equal to 0 and potassium is at equilibrium. So the arrows here are going in both directions and of equal size, representing that there is no net transport across the membrane. Note, under these circumstances, the two forces are counter-opposed to each other. We have the potassium concentration gradient driving potassium out of the cell from 100 millimolar to 1 millimolar. But we have the negative membrane potential attracting potassium into the cell. And these forces are working equally but in opposite directions. If the cell were to change its voltage to minus 30 millivolts, and we'll get into that in a moment as to how a cell could do that, this would cause an increase in the driving force inward into the cell by increasing the attraction of potassium into the cell. Thus, if we do a calculation here, we would find out that potassium uptake would be passive, moving down its chemical potential gradient because the electrical gradient is stronger than the opposing concentration gradient. And if we were to change the membrane potential to minus 75 millivolts, a more positive value, and there was still potassium uptake occurring, then we would say that transport had to be active. The uptake was an active transport process, since the electrical gradient is too weak to attract the potassium against its concentration gradient, and energy must be put into the transport process in order to move it from a lower chemical potential to a higher chemical potential. So there's an easy way to determine active versus passive transport by using this simple equation. If we compare the two chemical potentials, the inside of the cell versus the outside of the cell, by subtracting the chemical potential of the outside of the cell from the inside of the cell, this is equal to ZF times the membrane potential, which was measured, minus the Nernst potential, which is the calculated equilibrium membrane potential. We can come up with a sum here as an example. Let's go through this calculation for the minus 75 millivolts. So we have a charge of +1 times F, which is 96.5 joules per mole per millivolt, times the measured membrane potential or actual membrane potential at minus 75 millivolts minus the Nernst membrane potential at minus 118.4 millivolts. This is for a cell that has a concentration of potassium outside the cell of 1 millimolar and an internal concentration of 100 millimolar. When we make this calculation, it comes out to be 4,188 joules per mole. This means that the chemical potential inside the cell is larger than the chemical potential outside the cell. And if uptake occurs, then it would be going uphill and is therefore defined as active uptake. In addition, the cell will have to have used a minimum of 4,188 joules per mole to transport that potassium into the cell. For the case where the membrane potential is minus 130 millivolts, We go through this calculation again, and we find that the calculation comes out with a value of minus 1,194 joules per mole. Under these conditions, uptake would be passive, since this shows that the chemical potential outside is larger than the chemical potential inside because of the negative value. The minimum energy to pump potassium now out of the cell against its electrochemical potential gradient would be the 1,194 joules per mole.
So are ions actively or passively taken up? In 1967, Higginbotham et al. Attempted to answer that question by measuring the electric potential in the roots of a plant and measuring the ion concentrations internally and externally. What did they find out?
Well, Higginbotham et al. In 1967 attempted to make these calculations in the days when there were cruder instruments to make these measurements. So they had a number of ions in solution outside of the roots, consisting of potassium, sodium, magnesium, calcium, nitrate, chloride, phosphate, and sulfate. And the outside external concentrations in the medium are listed here, ranging from 2 to 0.125 millimoles per liter, or millimolar concentrations. They then measured the membrane potential inside the cells of the roots and came up with an estimate of minus 110 millivolts.
Now, this estimate of the membrane potential in the roots was somewhat inaccurate due to the cruder technology that they used in those days. It was not feasible to have a microelectrode that we could stick easily into a root cell, let alone a root tip cell. Therefore, the microelectrodes most likely went into the vacuole, and the membrane potentials that they measured were most likely the sum of the membrane potentials across both the plasma membrane and the tonoplast. We now know that the membrane potentials are lower than minus 110 millivolts. They could be in the range of minus 150 or even as low as minus 180 millivolts. So we also know that the membrane potential difference between the cytoplasm and the vacuole across the tonoplast is about plus 30 millivolts. So most likely these measurements in the minus 150 range. So these calculations are based upon the minus 110 millivolts. Those calculations will be reasonable in our conclusions that we'll draw here. But in fact, they are cruder estimates and not as precise as they could be if they had made these estimates at a different membrane potential. In fact, the differences might even be more extreme.
Likewise, since these cells were relatively mature cells, the vacuole makes up most of the space of the cell, and the concentrations of ions that they measured in the root cells were in fact probably more on the average of the ion concentrations of the vacuole, which in some cases is quite different from the cytoplasm, as I'll explain in a moment. So let's go through the calculations here for a moment to determine whether or not transport was active or passive for these ions. So by looking at potassium with an external concentration of 1 millimolar, it was predicted by the Nernst equation that it should have a concentration of 74 millimolar inside the cell. And in fact, they observed 75 millimolar. Now, you could argue this is slightly more, but in reality, this is probably not significantly different, and therefore potassium appeared to be at equilibrium. However, when we look at another cation, sodium, the same concentration outside, its prediction therefore would be 74 millimolar inside the cell. It was found to be only 8 millimolar. This means that the cell was somehow able to exclude the sodium or transport it back out of the cell to keep its concentration very low. So the concentration gradient is much stronger than the electrical gradient, and transport of sodium into the cell was a passive process. Magnesium was at 0.25 millimolar, and therefore with a +2 charge, it had a much higher predicted concentration of 1,340, which is extremely high and would probably kill the cell. And the actual observed concentration was 3. So again, there's a strong driving force for passive uptake of magnesium. Calcium likewise had a higher concentration at 1 millimolar in the external solution, and it would be 5,360 millimolar.
That's a concentration of about 5 molar. That's a concentration that would be impossible for calcium to occur because calcium easily precipitates out with sulfur or phosphorus at much lower concentrations than that. And in fact, those concentrations would be lethal to a cell. We now know that calcium is kept at much lower concentrations to avoid these toxicities in the nanomolar range. And as a result, we can see that the measurements here that are estimated to be 2 millimolar are still much higher than what it would actually be in the cytoplasm, supporting my earlier argument that they are most likely measuring vacuolar concentrations, as calcium concentrations in the vacuole are known to be in the millimolar range.
But it points out, again, that there's strong gradient for passive transport into the cell. The opposite is true for anions. If we look at nitrate at 2 millimolar in the external concentration, the Nernst equation predicts that we would have 0.027 millimolar, or 27.2 micromolar, inside the cell of nitrate. In fact, what was observed was 28 millimolar, so much higher concentrations inside the cell, indicating that nitrate uptake must be an active transport process since it's going against its chemical potential gradient. Note that nitrate has to be transported against its concentration gradient, and since there's a negative membrane potential, it's in fact repelled by that negative charge on the inside of the membrane. And so it's going against both forces, and this is fairly clear that this is an active transport process. The similar thing is true for chloride, for phosphate, and for sulfate. So what can we conclude from these results? Anions are actively taken up across the plasma membrane in plant cells. Potassium may or may not be taken up actively. Other cations are taken up passively. We can also see that there are differences between the cations and the anions, indicating that uptake is selective and not simply driven by concentration gradients and the electric potential. They also did some measurements on another species of plants and found that there were different values for that plant, indicating that there is genotypic diversity in these traits. So how is ion uptake regulated? We're going to go through several examples of transport mechanisms here. Note on the right-hand side, in the conditions that we're showing here, we have a difference in chemical potential. That is, there's a high chemical potential on the upper side, a low chemical potential on the lower side. So we have a chemical potential gradient going from the outside to the inside. So if we look back on the left-hand side, we can see an example of simple diffusion occurring where the transport of an ion is being moved across a lipid bilayer without any transporters. As we spoke about before with water transport in the previous lecture, can happen because of the fluidity of the membrane, causing openings where every once in a while an ion can slip through. It doesn't happen very often. It's rare, but it can happen and does happen. And eventually the ions will cross a membrane and reach equilibrium if we had two compartments separated by a membrane. In the other example here, there's a channel protein. Like the aquaporins or water channels that we discussed before, there are channels that can move ions across a membrane. Basically, it's a protein with a pore going through the center of it, which allows the ion to pass across the membrane when it's open. This is a passive transport mechanism, always. Channels always have passive transport. The ions move down their chemical potential gradients through a channel. In the other example here, we have a carrier protein. And this mechanism is not as clearly defined in plant science yet, but we do know that there are carrier-type proteins in plant cells. And this carrier protein, by some unknown mechanism, moves an ion across the membrane through the protein, but not as a channel. Another kind of transport protein is a pump. That is, it transports an ion with the use of energy across the membrane against its chemical potential gradient, as shown here. So we can call this a primary active transport mechanism. There are something that we'll talk about called secondary active transport mechanisms in a moment. Let's look at a generalized structure of a potassium selective channel. And I say potassium selective because it is relatively specific for only potassium moving through this channel and not letting other ions through it, through special fitting of that potassium channel through the pore. In A, we're looking from the end down onto the membrane, and we can see that these helical coils are surrounding a pore, and the potassium is fitting through that pore. On the right, we can see from a lateral view through the membrane of a diagram showing that the protein is made up of 6 membrane-spanning domains which form around the pore, and that there's an N and a C terminal on the cytoplasmic side. Important to note because these can be regulatory in some cases for certain channels. Also, what's interesting to see is that the S4 membrane-spanning domain has a charge to it, and this makes the channel sensitive to the voltage. So it's a voltage sensing region, and should the voltage change, it can cause a structural change or conformational change in the protein, causing it to move around, which may open or close a pore.
So what generates a membrane potential in plants?
First of all, we have the diffusion potential, as I've described already, which is created by the differential permeability of ions across the membrane. This can represent maybe minus 40 millivolts of voltage differences between the membrane, but the majority of the membrane potential is driven by electrogenic pumps. These are proton pumps in plants. In animals, they have sodium-potassium ATPases, but in plants, we have proton pumps or proton ATPases, which are located in multiple membranes in both the plasma membrane and the tonoplast. They create membrane potentials by pumping protons across the membrane, creating a charged difference.
As the positively charged protons are being pumped out of the cell, it is removing positive charges from the inside, making it more electronegative, and making the external side more positive, and increasing the proton gradient on the outside towards the inside.
They require ATP to drive these protons across the membrane against their electrochemical potential gradient, as we can see here. So on the left-hand side, at the plasma membrane, we can see there are some proton pumps that are utilizing ATP to pump a proton from the inside of the cell to the outside of the cell into the apoplast. And this requires energy since the pH outside the cell is a pH of 5.5, and the pH inside of a cell is typically around 7.5. So there is a 100-fold concentration difference from the outside to the inside, and those protons are being pumped against their concentration gradient. We also see at the tonoplast on the inner side of the cell that there is a proton pump pumping protons into the vacuole, and in this case it's pumping 2 protons. So it's extremely electrogenic, creating additional charge differences. And there's one other kind of proton pump that is known as a PPIase, which transports protons across the membrane as well. On the right-hand figure, we can see that we have identified the structure of one of these proton pumps on the tonoplast, and it consists of multiple subunits that form this proton pump.
The model for this complex protein is that the A subunits in the V1 part of the structure of this ATPase utilizes the ATP and causes a rotary motor action in the molecule, which is in the C subunits there that are rotating.
The point here is just to show you that we know quite a bit about these mechanisms and how they work. So let's talk about secondary active transport for a moment. This increase in concentration of protons on one side of the membrane versus another is known as a proton motive force. And in that is a stored energy. So we've used ATP to create that proton gradient, and that energy can be utilized to transport other molecules or other ions, as shown in this example here. So if we look at the left-hand side of the figure, figure A, we can see that the concentration of protons is much larger on the outside of the cell and much lower on the inside of the cell. And the arrow represents the gradient for this proton. And on the other side, we see S, a substrate, that is higher in concentration inside the cytoplasm and lower on the outside of the cytoplasm. So its concentration gradient is from inside to out. And in order for S to be transported against its concentration gradient, or in this case really the electrochemical potential gradient, it requires energy. In this example, we see a hypothetical protein with two binding sites, one for protons and one for S. And under circumstances where neither of the proton nor the S is binding to the protein, it takes a conformation which opens up a binding site available to protons on the outside of the cell. So randomly, by random kinetic energy, this proton will eventually bind to this binding site. And as a result of binding to this protein, it will cause a conformational change in the protein, seen in scene B, where once it's bound, it opens up and makes a binding site available for S on the outside of the cell only. The inside of the cell is blocked for either ion. Once the S and protons have both bound to their binding site, the protein will go through an additional conformational change, which results in the opening to the inner side and a closing on the extracellular side. And both protons and the S can leave to the interior side by random kinetic energy. Once both ions have left the protein, then the protein goes back into the original conformational state that it was at the beginning of this cartoon. And it is now ready to accept protons and eventually an S molecule. So it is the action of the proton moving down its chemical potential gradient that allows for this to operate.
So there are two types of proteins that can transport two ions or substances at the same time. They are known as symports or antiports. So these transporters can use both active and passive transport mechanisms depending upon the chemical potential gradients and the ions involved, as I'll explain here.
They can use this secondary active transport or proton motive force to drive transport of another molecule. So in this example on the left, in A, we have a symport transporter where the proton is moving down its electrochemical potential gradient from the outside of the cell into the cell, as we just described in the previous slide. And likewise, it drives the transport of A in the same direction, as we just saw in the previous example, an example of a symporter, where both ions are moving in the same direction against the electrochemical potential gradient of A in this particular example. So its proton motive force is being used to transport A against its electrochemical potential gradient. In the example on B, we have what is called an antiport, where the electrochemical potential gradient of protons is passively driving the transport of B against its electrochemical potential gradient. So there's movement in the opposite direction. So we have an antiport moving an ion against its electrochemical potential gradient, and we have a symport moving against its electrochemical potential gradient. Now, not all symports and antiports necessarily have to be active transport mechanisms. They could be happening in a passive way for both molecules if the situation or the circumstances will allow it. So remember, our definition of active transport here is always going to be the movement of an ion against its electrochemical potential gradient. That is the only definition of active transport. Just because it uses energy doesn't make it active transport. And this is a common misunderstanding where people say, oh, but it used ATP or used this other energy. that's not the definition of active transport by our definition in this course, which is a thermodynamic definition that I gave earlier. Okay, so let's look at a cell. And here is an example, very complicated slide on the left-hand side of many different kinds of transporters that we know now exist in the cell. We'll come back to this in a moment in a little more detail. But let's say it's broken down into pumps and symporters and antiporters and channels. And the cell also has a vacuole with a tonoplast, which also has a bunch of transporters that we know about that occur in these membranes. So on the right-hand side, let's just look at the tonoplast transporters for a moment. If we start at the top of the figure, we can see a number of examples of antiporters that have been discovered. Let's start on the left-hand side where we have a nitrate transporter with a proton in its antiport form. So the proton gradient, that is much lower pH inside the vacuole versus the cytoplasm, is the proton is going down its chemical potential gradient, but it's driving nitrate into the vacuole against its electrochemical potential gradient in this case. So it's causing nitrate to be taken up and stored in the vacuole. We know that there are also antiporters for sodium ions, where the sodium ion is driven into the vacuole. We also have calcium ions being driven inside the vacuole. And in this case, it takes 3 protons to drive in 1 calcium. So it could be electrogenic since there's only 2 plus charges. Cadmium is also taken up this way. Magnesium, hexoses or sugars, sucrose can be transported through transporters that use protons as antiporters. And we have even here on the right-hand side a symporter driving protons and sucrose in the same direction out of the vacuole. There's another class of transporters known as ABC transporters, and these transporters utilize ATP, and they can transport more complex molecules like anthocyanins, which is a big organic molecule, and that can be driven into the vacuole for storage. Calcium is transported into the vacuole by a calcium pump, and it's using ATP. So it's a calcium ATPase in this case that is driving calcium against its electrochemical potential gradient. Now we have a series of channels. There's a potassium channel that allows potassium out of the cell. We have other channels that can do both potassium and calcium. It's known as a slow vacuolar or TPC1 channel. Each of these are specific and different from each other. We have a calcium channel that allows calcium out of the vacuole and is triggered by IP3, a signaling molecule inside the cytoplasm. We have anion channels in the tonoplast that allow anions to be taken up into the vacuole. So that's moving down passively by diffusion. Remember, they move by diffusion in these channels down their electrochemical potential gradients. And then some cases, we have a fast vacuolar channel that allows cations, in particular potassium, and they can go in either direction depending on what happens to the electric potential. So the cell electric potential can change, and this can cause changes in the direction of the potassium transport down its electrochemical potential gradient. And then finally, we see 2 of the proton pumps, the PPIase and the ATPase in the tonoplast. Whew, it's a lot, huh? Well, there's a lot more than that now that we've discovered, but that's just some of the transporters. If we go back out to the plasma membrane, we can see, again, on the left-hand side, we can see the proton pumps. We have transporters that take up metals like iron, manganese, zinc, and cadmium. The exact function of these is not really clear at this point, how they work. We have a proton-driven nitrate symporter that is driving in nitrate. Remember, we found out that nitrate was being taken up actively. So it's using the proton motive force to drive nitrate uptake, as well as phosphate uptake, as seen in the next symporter. In some cases, we've identified a sodium-potassium symporter that also could use protons instead of sodium to drive potassium uptake. And we know that there's another symporter that is using protons that drives sucrose absorption into the cell from the apoplast. We'll learn more about that when we talk about phloem transport. There are transporters for amino acids to take up amino acids across the plasma membrane using protons. Peptides, which are sequences of amino acids of about 20 amino acids, so they're long chains of amino acids. And we have antiporters, such as the sodium proton antiport, which drives sodium out of the cell. Sodium is not a desirable ion for plant cells. It can become toxic. So it uses energy to pump out those sodium ions to protect the functions in the cytoplasm. And we also know that sucrose has an efflux carrier of some mechanism that allows sucrose to be transported out of the cell. Now let's look at the channels. There are multiple channels in the plasma membrane. We have a cyclic nucleotide gated channel. That means gating is a reference to controlling the opening and closing of that channel, and it requires a cyclic nucleotide to allow it to be opened or closed. There are inward rectifying potassium channels. I'm not going to get into rectification. And there are outward rectifying channels. There are anion channels, which transport malate, chloride, and nitrate out of the cell down their chemical potential gradient. There are channels that operate, as I mentioned, by depolarization. When the membrane is suddenly depolarized, it can cause an opening and allow calcium to move down its electrochemical potential gradient. And there are other calcium channels. When the membrane is hyperpolarized, will also open and allow calcium to enter into the cell down its electrochemical potential gradient. And again, the proton motive force is set up by the proton ATPase, and there's also a calcium pump that is driving calcium outside of the cell. Remember, the cytoplasmic calcium concentrations are very low at the nanomolar range, in the range of 50 to 100 nanomolar. The calcium concentrations on the outside are millimolar concentrations. So this calcium has to move down its electrochemical potential gradient in an inward direction because of this steep concentration gradient plus the negative electric potential that is attracting that calcium very strongly into the cell. So passively, calcium will move down its electrochemical potential gradient through channels, and it must be pumped out in order to keep the calcium concentration very low. The reason it keeps it very low is because calcium will become toxic to the cell in any higher concentrations.
So in summary, in this lecture, we've covered the molecular mechanisms for ion transport in membranes, and we've discussed the driving forces that influence that transport. So that involves the chemical potential, which includes both the concentration gradients of the ions involved, as well as the electric potential of the membrane. And it's the two of those, the electrochemical potential, that is important in determining what the overall forces are in driving that transport process, which may be active or passive, or even both in some situations, such as an antiporter. In the next lecture, we'll look at the kinetics of uptake and transport into plants, and we'll look at the pathways by which those ions move through the root and into the leaves of the plant. I hope you are finding this series useful and interesting. If you're a plant lover like me, I know you are. For those who are just gardeners, this may be a little bit too deep for you, but those who would like to understand how things work in this world, this is the lecture series for you. And if you like this series, then please like it on my YouTube channel so that other people can become aware of it as well. Have a great day.